Hello friends, welcome to the program Differentiation and Growth. Differentiation is the full sequence of changes involved in the progressive diversification of cell structure and functions, that is the authentication of development. From fertilized ovum, all cells arise by repeated divisions and inherit the same biochemical endowment, morphogenic assignments and same functions. Normally, the differentiation process is irreversible. Almost every cell of developing embryo multiplies and becomes different from the original condition as well as from one another. Cells of an intercellular structure as fibers in the connective tissue, bones and cartilage matrix, cuticle, etc. divide to produce visibly differentiated cells, showing morphological expressions of spatial functions of differentiated cells. In histological differentiation or cytodifferentiation, cells achieve biochemical and functional characteristics. Now let us talk about functional differentiation. Both undifferentiating and differentiating cells are capable of performing the processes of metabolism, amoeboid movements, and other reflex stimuli reactions. Beside these biochemical capabilities, which are superposed general functions, this process is called behavioral differentiation. For example, liver cells secrete bile. Now, chemodifferentiation. Chemodifferentiation serves as a trigger for morphophysiological differentiation and specialization of cells under the control of genes. Specific enzymes or specific combination of enzymes are the essential parts of the functional mechanism of a cell. Thus, it can be said that differentiation is the production of unique enzyme protein pattern. The initial step in the differentiation of cells is made by the cytoplasm, but the final stage cannot be achieved without the synthesis of enzymatic and structural proteins and takes place under the guidance of DNA, RNA, hormones and vitamins. Now, genic or molecular control of differentiation. Specific proteins are involved and elaborated by individual cells to characterize a particular diversification pattern. This phenomenon may be due to the fact that either genome of cells is different owing to the mechanism of differential replication or transcription of DNA if they are identical. Chromatin diminution. Boveri in the year 1899 observed chromatin diminution in a nematode first and found that from the cells closest to animal pole, part of the chromosomal material is spilled into cytoplasm during second cleavage. At the 32 cell stage, only 2 cells have the full gene complement called primordial germ cell, whereas the remaining ones have undergone chromatin diminution called presumptive somatic cells. Now, differential transcription of DNA. In the operation of selective transcription of DNA, either the total genome is initially active and individual genes are then selectively replaced, or the total genome is initially inactive and individual genes are then selectively activated for transcription. Let us talk about a regulation of differential gene action for differentiation. In eukaryotes, the functions of genes can be regulated anywhere between gene replication and the release of completed polypeptide chain from the ribose, as the synthesis of RNA from DNA is termed as transcription and the synthesis of protein from RNA is termed as translation. Therefore, gene activities can be regulated at following levels. Control of differentiation at the level of genome. DNA may be modified by alkylation or methylation reactions for which necessary enzymes are present within the cell. Methylation of any nucleotide basis of DNA has to be carried out anew in each chromosome once reaction has been completed. Due to increase or decrease in the DNA molecule quantity, control of differentiation occurs, in which extra amount of DNA somehow regulates the protein synthesis for differentiation. In Xenopus, each normal cells contain repeated gene sequences, about 900 copies of the genes for ribosomal RNA, which are called as reductant genes. During oogenesis, an amplification of ribosomal RNA takes place, so that size or number of nucleoli is greatly increased and large number of ribosomes is formed. Sometimes depletion of genetic information due to deletion of genes from one of the chromosomes result to mutant of animal gene. Now, let us go to the control of differentiation at the level of transcription. The first is gene regulation by histones. Double-stranded molecule of DNA has free acidic groups of phosphoric acid on their outer surface, and this can establish firm bonds with the amino groups of the basic amino acids of histone chains. This intimate association of DNA and histones prevent the DNA from interactions with other substances in cytoplasm, thus serving as templates for RNA production. Histones inhibit DNA primed RNA synthesis to diminish DNA polymerase activity. Thus, histones serves as repressors. Now, gene regulation by acidic proteins. These are non-histone phosphoproteins with tryptophan and tyrosine as the main constituents. These proteins remain intimately associated with DNA, called histone free complex, and are considered as more vital for gene regulation histones. The DNA histone complex remains inert to the transcription, so that acidic proteins interact with basic histones, putting the histones of certain critical genes as promoters, so that genes can be transcribed. Now, gene regulation by heterochromatinization. Heterochromatin of interphase has some specific role in gene regulation. For example, synthesis of proteins is very less in human beings, where blood cells contain large masses of condensed heterochromatin, while in white blood cells, synthesis of protein is very less due to the lack of condensed heterochromatin. Now, control of differentiation at the level of translation. During translation, message carried by the messenger's RNA has to be decoded, and required amino acids are to be picked up to form various proteins, hormones, enzymes, etc., to bring about differentiation by influencing the path of protein synthesis. Post-transcriptional regulation mechanisms exist at several steps that are involved in the formation of proteins. Hormones are found to be more effective to induce differentiation at translation step, and they might facilitate diffusion of particular messenger's RNA out of the nucleus, stabilize their lifespan in the ooplasm, increase the frequency with which they associate themselves with ribosomes and become translated, and so on. Hormones may influence enzyme activity at translation level, or produce secondary effects, or affect chromosomal gene activity by getting localized in the nucleus. During hormonal stimulation of differentiation, mitosis precedes terminal differentiation process. Now, let us talk about levels of differentiation. The 4 levels of differentiation are: undifferentiated state is one. It consists of conversion of the undifferentiated cell to a cell with pancreatic character. At this stage, even before the onset of histogenesis, specific pancreatic proteins are present at detectable level and prove that this event may uncover all the genes required for complete pancreatic differentiation. Second is proto-differentiated state. This event involves conversion from the proto-differentiated state to the differentiated state. Cells undergo extensive proliferation which ceases in those cells which undergo differentiation. Synthesis of ribosomal RNA, assembly of functioning ribosomes, or development of the endoplasmic reticulum are the major events in this state. Third is differentiated state. It involves a mechanism that makes the cell definitely distinct from others. Alterations in specific enzyme activity among differentiated cells can be observed. Modulation of the differentiated state includes changes in the production of specific messenger RNA and changes in the ribosomal efficiency in protein synthesis very late in development. Modulation occurs in response to extracellular factors like hormones. Differentiation of stem cells is almost similar to the various levels of differentiation. Mitosis plays an important role for growth and also for differentiation. Stem cells. Stem cells are undifferentiated cells which act as a source material for specialized tissues. These cells appear in the embryo and remain throughout the life of the animal. Ultimately, act as precursors for the specialized tissues. Stem cells get differentiated with a shift in the developmental potential from the totipotent to more restricted condition. Erythropoiesis provides a good example of stem cells where almost identical levels of differentiation occur. It includes a sequential series of stepwise changes in which a pluripotent mesenchymal cell receives a stimulus and directs itself towards an erythrocytic series to form the precursor stem cells called the hemocytoblast. This process is called as determination. Now, the hemocytoblast becomes a proerythroblast. This proerythroblast follow 5 steps called maturation stages and can be directed as proerythroblast is a stage of active RNA synthesis and appearance of new proteins along with 4 cell divisions. Morphological maturations takes place in 6 definite stages in which multiplication, growth and synthesis of hemoglobin takes place. Basophilic erythroblasts are characterized by chromosomal condensation, reduction in nuclear activity and hemoglobin production. Third is polychromatophilic stage is an important stage in which synthesis and accumulation of hemoglobin increases and level of RNA synthesis slowly decreases. Orthochromatic erythroblasts are cells which possess inactive nucleus and cytoplasm filled with hemoglobin. These are incapable of cell division. Next is reticulocytes, ripen after 3 days of cell divisions and differentiate into red blood cells filled with hemoglobin. During this stage, cells expel their nuclei in mammals. Now, let us talk about tissue interaction during differentiation. In early amphibian embryo, differentiation of epithelium depends on its interaction with mesoderm. When it is isolated without mesenchymal cells, embryonic epidermis loses arrangement of epithelium, ultimately leading to cell degeneration and death. With mesenchymal cells, epithelium is preserved and due to healthy ectoderm differentiation, normal skin epidermis development takes place. Synchronized development and intimate relations of mesenchyme and epithelium suggest an interacting mutual control system. Sometimes Morphogenesis of an epithelium is modified by the mesenchyme with which it is grown, producing an epithelium that reflects the origin of the mesenchyme. Thus, it can be stated that the cells and tissues tend to acquifer locations according to the character of their individual or collective differentiation, or cells and tissues tend to differentiate according to their location in the organized system. Now, role of microenvironments in differentiation. The basal layer of cells in the epidermis consists of mainly proliferating cells which lose their attachment to the underlying basement membrane and crowd into superficial layers of epidermis to synthesize keratin. These basal layer cells are like stem cells which differentiate in response to any change in environmental conditions. Secondly, these cells have to be exposed to vitamin A so as to modify them into mucoid secreting epithelium. Thus, microenvironment of cells plays a major role in fixing the direction of differentiation taken by an unspecialized cell. If we place two cells in a minute hanging drop of nutrient culture medium, one of them gets differentiated. Slowly, the other cells receives cues from neighboring cell and starts the process of differentiation. Developing systems normally increase in size at least during part of their development. Growth is a general term used to describe this phenomenon. It comprises two main aspects: increase in cell numbers by cell division and increase in cell size. These two processes may in some examples occur quite separately from each other. For instance, cells in certain rapidly growing tissues, for example, the connective tissue or blood forming systems in vertebrates, may increase greatly in number, while the cells remain approximately the same size. Alternatively, in some organs, for example, the salivary glands of insects, the cells may increase greatly while remaining the same in number. Each cell becomes enlarged or hypertrophied. In such greatly enlarged cell, there is often duplication of the genes involving an increase in the DNA content of the nucleus, although no cell divisions takes place and the nucleus continues as a single body, although with a multiplied or polyploid set of chromosomes. In very many cases, however, the growth of an organ depends on increase both in cell number and in cell size. One case that has been well studied is the size of the wings of the fruit fly Drosophila. The number of cells in the wing can be easily determined, since each bears a single hair that can be seen and counted in simple microscopic preparations. It has been found that there is an accommodation of factors. If there is an unusually large number of cells, these may be somewhat smaller than usual, so that the total size of the wing remains relatively unchanged. After their initiation, the various organs and regions of an organism may increase in size at different rates. Such processes of differential growth will change the overall shape of the body in which they occur. Processes of this kind takes place very commonly in animals, particularly in the later stages of development. They are of major importance in the morphogenesis of plants, where the overall shape of the plant, the shape of individual leaves, and so on, depends primarily on the rates of growth of such components elements as the stems, the lateral shoots, and the vein and intervein materials in leaves. In both animals and plants, such growth processes are greatly influenced by a variety of hormones. It is probable that factors internal to individual cells also always play a role. Although Differential growth may produce striking alterations in the general shape of organisms. These effects should probably be considered as somewhat superficial, since they only modify a basic pattern laid down by other processes. In a plant, for instance, the fundamental pattern is determined by the arrangement of lateral buds around the central growing stem. Whether these buds then grow fast or slowly relative to the stem is a secondary matter. However, striking its results may be. To summarize the topic, it can be said that development always involves growth and differentiation. Differentiation is the process by which cells and tissues acquire certain characteristic features and become different from each other. Differentiation occurs simultaneously with growth, and the two processes cannot be separated. The embryo up to the formation of organ rudiments is very small in size. Growth is the increase in size. This is achieved by increase in the rate of cell divisions involving fresh synthesis of nuclear material and protein synthesis. All the organ rudiments increase in mass and the embryo attains the size and the shape of the adult. So, this is all about the process differentiation and growth. Thanks.